So the next two speakers will share a 30-minute slot. The first is Zina Cinker from Puzzle X in Barcelona, who will be talking about graphene. Both these talks will be about graphene as sensors. And there's Zina. Hey, good to see you. And take it away, please.
Oh, fantastic. I'm early to the party. Aren't I? Can I just take it on?
Can you what?
Can I take it on? Because I was supposed to be on in 10 minutes, but if you want me to take it on right now, I can go ahead.
Yes, please do. We're trying to beat a hurricane, but take your time. But yes, please go ahead.
Okay, fantastic. Well, thank you so much, Stu, for inviting me. And hello, everyone. I'm so sorry I was not able to be with you in the event. I would like to share my screen and see if I can pull up the presentation. Hold on one second. Let's do this. Bear with me. All right, we're going to go into presentation mode. And I am going to assume that we are going to be able to see my screen. Okay, if you cannot see the screen, please let me know. Somebody let me know, and I will try to troubleshoot. But hello, everyone. My name is Zina Cinker. I have to do a big disclaimer here, which is, although Stu Hemroff was kind enough to invite me to this event, I am not in the field of consciousness. I'm a condensed matter physicist, and I met Stu a few months ago in Sicily at Termina at the Science of Consciousness event, and we had some great conversations about the overlap between condensed matter, especially in the case of graphene and two-dimensional materials. One of the things that I brought up to Stu was that there are lots of development in this field in the past 10 to 12 years, especially in the area of biological interfaces with graphene and other two-dimensional materials that have been developed that I do believe that are going to have a direct influence in the science of consciousness studies. And what we wanted to do was, through this talk and also me inviting Carolina and sharing the stage time with Carolina, who is the CEO of InBrain's Microelectronics, which is one of the most prominent companies in the space of graphene and brain interfaces, is to make that bridge between these two different fields, because I believe that there are lots of learnings, there are lots of testing methods that could actually be aiding both sides. So the way that I will structure this conversation is that I'm going to first go over a little bit about what graphene is, a bit about a two-dimensional family of materials, talk a bit about what Stu asked me to speak on, which is the electron-phonon dynamics in graphene, and then I'm going to explain a little bit about something that's the field that we are very interested in Puzzle X, which is the synthetic biointelligence and the organoid intelligence community, which I do believe that probably are very much present in this event, but in case they're not present, I hope that this acts as a bridge so that they would be invited in this community, because I do believe that lots of exciting development is happening in that space of organoid intelligence, especially in the past year and a half that is happening. Starting with graphene, I think that probably most of you have heard about graphene. Graphene was a big buzzword in the past 10, 15 years. Graphene is one single atomic layer of carbon. It's a hexagonal lattice, honeycomb lattice structure, which is sp2 bonded atoms of carbon. And it is the first two-dimensional materials that we actually ever stumbled upon. It probably was isolated and exfoliated throughout the history many times. We just didn't know that it was there. It was theoretically conceived years and decades ago, but the consensus was that something this thin at the two-dimensional atomic layer would not be stable, so it was really never experientially actually looked after, experimentally designed. So in 2004, two physicists from Manchester, Andrei Gaim and Kostya Novoselov, exfoliated graphene from a piece of graphite, which was, if you've heard it in the news, it was the famous Scotch tape method, which is they cleave the graphene, basically. What you're seeing in this video is that graphene is one atomic layer of carbon that is cleaved off of a piece of graphite. That's basically what it is. They use a piece of graphite and they use Scotch tape to peel off these different layers. And they would put this thing back on and they would peel it off again and again and again until they ended up with one single layer. And that is what they call graphene. And they won the Nobel Prize for Physics in 2010, very short, 6 years later, after this isolation, which speaks to the importance of the discovery of the first two-dimensional materials that we've seen. Graphene has a very exotic electronic and optoelectronic properties, which makes it very unique for the sensors and also optical sensors and different kinds of sensors, biosensors. And I bet that Carolina can be guiding us through this a lot better than I can, since my field was not interfacing with biology much. There are many exotic properties from plasmonic, thermal, mechanical. Graphene is 200 times the tensile strength of graphene in the planar is 200 times stronger than that of the equivalent weight in steel. It is at one atomic layer, it has a very small absorption in the visible range, which makes it almost transparent. It is extremely flexible, and electronically it exhibits some very exotic properties, which is electrons in graphene, which is a semi-metal. Can travel with the speed of light in crystalline structure, which gives it electron mobilities higher than anything we've ever seen before. Very, very attractive electronic-wise, very attractive optoelectronic-wise, and also in terms of thermal and the phononic dynamics that we're seeing, very interesting material, which opened the path for a lot of different developments. Some of those developments were in the structural realm with mechanical strength, where we have seen a lot of development coming out. Many of them were in the electronic sector, and many of them are these days, most of them are in the optoelectronic sector, which we are seeing a lot of development in. The thing is that graphene, although we first discovered graphene and we say, okay, this is the first one, graphene was not the first one. And it's since then we have discovered many of them, thousands of different heterostructures that are identified: graphene, molybdenum disulfide, MOS2. There are many different ones that are coming out and discovered more and more. And the beauty of these heterostructures is that now we've got like Lego pieces of two-dimensional materials with very extraordinary properties, or call them superheroes of the materials world, basically, that are going to be able to be layered on top of each other to create compact properties and compact structures, unlike anything that we've ever seen before. And it is truly fascinating what has been done in the past 10-15 years in terms of developing different structures with these nanomaterials that are 2D and planar structures, especially with these van der Waals heterostructures that we're seeing right now in this picture. And the work continues to go on, and it has opened up a very exciting kind of prospect field in condensed matter and stacked up electronics. And for the purpose of this field, for the purpose of this conversation here, we're going to be focusing mostly on the field of bio, and Carolina again will be our guide in this conversation. So what Stu asked me to speak on, which is the graphene electron phonon dynamics. First of all, I have to say, I don't know much about biology. I am not in the field of cognition. I am not in the field of brain. I've been always fascinated by it. And one of the reasons why I was in Taormina at the TSC a few months ago, where I met Stu and many of the leaders in the field, was because we, as the Puzzle X community, which we have an event in Barcelona every November, we're very much focused on some of these areas, such as quantum computing, such as condensed matter, such as brain interfaces, etc. But we don't look at consciousness. And this was the first year that I got the approval of my board to actually introduce the topic of consciousness. So that was why I was socializing with the consciousness community to understand what are the overlaps and also what might be the current bridges that could be built between these fields. When I spoke with Stu about electron phonon dynamics of graphene, I never knew that there might actually be an overlap. So I'm going to speak about kind of what the work that I've done scientifically in my background is. There might be overlaps or there might not be overlaps. Whatever I say might be a reach, but the intention was here for us to talk about this and explore if there might be something that is going to be opening up the field from both sides to this topic. I do not know any, because these electron dynamics I'm going to be speaking about, especially in the pump probe spectroscopy and femtosecond ultrafast spectroscopy regime, are in the femtosecond time window. I do not know if, besides the photosynthesis, if there are any cognition-based or any brain-based interfaces or phenomena that are happening that are within that time scale. So if anybody knows about that, or if anybody would like to talk with me about this, please do reach out to me. I can be reached on LinkedIn very easily, Zina Cinker. I would love to understand if there are biological phenomena specifically in the cognition realm that happens in the femtosecond time regime that could be used by ultrafast spectroscopy, especially in a pump probe technique, that could decipher some things. And then we could build that bridge between these two different communities. From what I have seen from my side, my field does not interact much with the biological matter, and I would like to see what can be rectified in that situation if there is any overlap. I do not see the audience in this field, so my apologies again for not being with you. But in case there needs to be some background information on femtosecond pump probe spectroscopy, allow me to go spend a few minutes going over some basics of pump probe spectroscopy. Ultrafast pump probe spectroscopy is a very old and tried technique, and is quite precise. What we're doing is that we're dealing with pulsed lasers of femtosecond timescale. Femtosecond, if there are folks in the audience that are not familiar with that, is 10 to the minus 15 of a second, is million times a billionth of a second. It's a very short time window in the beginning of the process, especially in this case. What we're going to be talking about is the pumping of electronics properties and excitation of the electrons to the conduction band. It happens in the femtosecond and the picosecond time regime. Again, it's a very, very fast processes, and that's where it says ultrafast spectroscopy. And what happens is that we have a very strong pump laser beam. We're using two laser beams in this pump probe spectroscopy. One laser beam is going to go through the sample. In this case, we were studying graphene. It goes through the sample. It excites electrons to a certain conduction band. And what it's going to be doing, hey, Carolina, I think we've got our... Hi, Carolina. I'm still doing the basics of graphene, and then I'll hand it to you in probably 5 minutes.
Very good.
Fantastic. So what we're doing in the pump and probe spectroscopy is that we are pumping the electrons with the energy of using photons with the laser energy, and then we're sending a much weaker probe signal, which is going to go through the surface. It gets bounced off at many, many different moments, as you're seeing in this temporal time scale that you're seeing here in the delay times. It goes up and then it recovers. And what it does is that it's giving us a snapshot and a picture of the electronic properties in the conduction band, or which is actually discovered, or we can deduct that by looking at the transmission or reflection of the sample that we have. Lots of jargon, lots of things that's probably not relevant. All of that to say that we've got extremely fine techniques. And I know that femtosecond lasers are used in surgeries, are used in many different techniques in biology. In this instance, we're using a pump and a probe to look at very fast timescale phenomena happening the moment that a photon is hitting an electron in a substrate like graphene. What happens to that photon? What happens to that electron? How is that energy that is transferred into that electron, which goes into the conduction band, going to be relaxed into different phenomena? What we have studied is to show how many different channels of relaxation exist within that phenomena. And what we discovered was that at the time that we were doing the study, most of the studies were done on suspended graphene, where it had no interaction with the substrate beneath. So all the phenomena were happening in a place that was isolated within the planar structure of graphene itself. What we did was that we had graphene on top of different substrate, and we studied that, and we found out that there are very strong coupling between the electrons of graphene, excited electrons of graphene, and the phonons of the substrate itself. So what we found out was that one of the relaxation mechanisms that allows the electrons in graphene to relax much faster when it is on a substrate is through this coupling through the substrate itself, using the surface phonons of the substrate. some of you might be thinking, okay, duh, of course there's going to be interactions between a layer of graphene or anything. If we had, let's think of an aluminum foil, we put it on a box and we hit it with laser and it's heating up. You would say, is there going to be interaction with the surface, with the box itself? Of course it's going to be. But what I want you to remember is that those phonon interactions that results in heat happen much, much later time scale. Here we are talking about the first few femtoseconds of a structure, which is extremely short period of time. What we've discovered is that there are interactions with the substrate, not only laters through the assistance of regular and conventional phonons, but there are surface phonons and optical phonons within the graphene, intrinsic optical phonons within graphene that happens. All of that, what does that mean? That means that the graphene electronic dynamics can actually be tuned based off of the substrate that it's sitting on. How does that relate to biological matter? I absolutely have no idea. I do not know if femtosecond structures or femtosecond time windows are relevant in that time scale for anything that could be studied. But in case they are, and that's where my curiosity comes in case there are and they are relevant, then these pump and probe spectroscopy with a surface with graphene might be a window to us understanding how these electrons relax within graphene and might give us a window in terms of the energy transfer between different structures and might give us a little bit of more of an information about the model that these structures are using, whether or not there are neurons. I don't know if there are the scales that could be studied or not. This was an open conversation that we had with Stu, and he graciously invited me to talk about this a little bit more. And I'm going to be handing it to Carolina in a few minutes. I just want to go over very quickly some other areas of interest for us that we cover at PuzzleX around this idea of the intelligence. And again, I do believe that hopefully the synthetic biointelligence community, organoid intelligence community is present in California right now. If they are not, I encourage everyone to look at some of the seminal work that has been done in the past few years. One of them led by Cortical Labs from Melbourne, where they looked at the first what they call dish brain systems, where they looked at a closed loop system. They grew the mouse cortical neurons on top of an array of chips, of microelectronics and silicon chips. And what they showed was that there is an input and output, and there is actually a very strong learning mechanism that can be induced in these systems. What we did at PuzzleX was, let me see if this video plays. What we actually did at PuzzleX in November was that we did the first live demonstration of this phenomena, where we connected from Barcelona on the stage to Australia to connect with a bunch of brain cells sitting in a petri dish, and that is Han Wang, who is the CEO of Cortical Labs, and we showed that there is a learning. You can see right now that this dish brain is actually learning live to play the game of Pong here, and you're going to see kind of the fingerprint of the neural activity and there, which was quite exciting and fascinating to see this. We didn't know if it was going to work or not, but it did work, and it was wonderful to see that in person as well. The continuation of this field, which I am very excited about, is culminated in the work of Thomas Hartong Lab from Johns Hopkins University. I'm sure many of you have seen this work coming out in the past year. Since February, there's been a lot of media coverage for this work with the organoid intelligence community. Now they are looking at 3D structures of these brain, basically like infrastructure, which is the continuation of this brain in a dish. And there's exciting things that are coming out of this this community as well, which we're going to be covering in November at PuzzleX. With that, I would like to thank again Stu, and I would like to thank the community for inviting me as a condensed matter physicist. Amazing to be able to give a talk here, and I am very fascinated by the field. I can be reached at zina.cinker@puzzlex.io, or you can find me on LinkedIn. And I'm going to now pass the microphone to Carolina. I'm going to take my presentation away and also mute myself and take my video away. I look forward to hearing from all of you. Thank you so much. Carolina, the floor is yours.
What a great introduction. Thank you, Zina. Let me share my screen. Hope you hear me well.
We can hear you. We hear you okay.
Very good, thank you. Do you see the screen well as well? Yeah?
Yes,
All right.
we see you. We hear you and see you. Great, thank you.
Thank you. So I'm going now to explain how we use graphene to understand the brain and treat the brain. And what we know is that after decades of clinical work, this is how much we know about the brain today on the left, which is the size of a zebrafish brain, which is about 100,000 neurons. However, the human brain, as you well know, is nearly 100 billion neurons. And we need to understand it because we need to treat it, since 1 out of 3 people have a neurological related disorder. And as you might know, 30% are refractory to pharmacology and therefore don't have much solutions. And as Zina used to say, every leap in humanity has been linked to a key material. So from Stone Age, today we're on the Silicon Age, we believe that graphene represents this breakthrough in the advancements of neurotechnology. I'm not going to say much again about graphene. You have seen from Zina's material, what is it about? I'm just going to showcase how we use it. And the most important is why we decide to use graphene. So the problem is that current neuromyoelectronic therapies, we think they are lagging behind. There's very low adoption after decades. And in fact, metals are only able to produce quite bulky systems. And in many cases, 50% actually reject these systems. Then when we try to understand the brain, the tools that we have today are also metal-based, and they are big. They are poor resolution, they're low count, and as a consequence, we rely on the precision of the neurosurgeon to put those big dots into nuclei or targets of the brain, impacting patient-to-patient reproducibility. And most of them can only stimulate. So they are not personalized. They cannot record brain activity. Instead, lately, with the brain-computer interface booming, I think that's changing. But there's nothing commercialized that actually is bidirectional. And that's what we are changing here. So first of all, graphene can miniaturize to a cell-like size. And that enables also the coupling with modern electronics and therefore much more and efficient systems, implantable systems in this case. The interfaces that we can develop are bidirectional, they're flexible, and they have ultra high density and resolution, and therefore they allow us to understand better what's going on the brain, but also create a feedback response in closed loop live for every particular patient. And that's where we are heading, towards precision neurology, where actually we could, based on that decoding of the signals, create a response, but also predict what's going to happen to that patient based on artificial intelligence algorithms. We have a graphene that we call Ignite, and what we are doing is actually 2 things. So we are aiming to generate better outcomes because we can have 10 to 100 times more precision via the undercovering or decodering of a broader range of biomarkers at higher density and resolution, and then increase also the patient adoption because of that reduction of size, but also power requirements, thanks to the properties of graphene. So we can inject 200 times more charge injection at a very low impedance and also have a very high signal-to-noise ratio. The way we are doing is looking at circuits. So the brain has been decoded in the last decades, nuclei by nuclei, and we think that we'll never make it if we continue like that. So we are going pathway by pathway or circuit by circuit. And this is just an example for Parkinson's disease. Now, this has been done in some way when we look at the subthalamic nucleus exploration, but has never been done in a circuitry manner. And also never has been done in high density and high resolution. So we believe that by doing that in such a resolution and with graphene, we can increase therapy effectiveness, and also we can help improving the life of the patients that are impacted by several of these diseases. Parkinson's is just an example, But we are looking at a speech impairment by speech synthesis, more on the brain-computer interface side, and other indications. Now, because you are looking very deeply into consciousness, the closest thing that we can relate to cognition is by looking at the relationship between motor function and cognition. And if we look back in time, this association between motor function and cognition can be very well understood if we see the evolution of humans from 4 legs to bipedalism, and how the circuits that connect the cortex, which is a massive learning center, towards basal ganglia and cerebellum, have learned over time to automate activities. And the sophistication of these actions have led to a very efficient brain. So if we can understand this, and we can actually understand how these circuits work, how they are modulated, we could probably in the future create also a more effective brain if we are able to predict how to delegate that automation or how to help the brain to automate. And the way we are starting to think about this is in the case of Parkinson's disease, the subthalamic nucleus has a huge implication in the modulation of the motor cortex. And over the years, we have learned that the subthalamic nucleus is actually a very complex nucleus with a very specific somatotopy, with a premotor area, a motor cortex area, associative and limbic areas. And right now we are targeting, the only commercial interface, brain or deep brain interface that is available is this deep brain stimulation. However, deep brain, again, is low resolution, low count. So at the maximum, 1 or 2 contacts fit into these nuclei, and there's not a specificity and understanding of each of these regions. Now, we also know that the subthalamic nucleus connect to the motor cortex or to the cortex, let's say, for different functions. So in this case, there's this paper from cooperation between Oxford and California, where we're looking at how actually when humans are moving, they also, when they have alarming signals or signals where they should stop, they have to actually raise the level of the threshold of decision making, and then rethink what are they going to do to move again. And that's kind of what we are doing also when we are trying to control Parkinson's disease. In this case, the stopping action is generated by the right frontal gyrus that actually communicates with the STN and the GPI, and then back to the premotor cortex regulating, or in this case, stopping movement, and then raising the threshold for decision making, and then understanding what that person should do. And in case of conflict, there's also a very similar path. Now, these pathways are regulated by brain signals. So in this case, beta correlates more to the stopping, and low frequency oscillations to the conflict. What we are doing here is studying these correlations at a very high resolution and with a lot of, let's say, more information than before. And just as an example, what we've been able to achieve in the last years is looking at the resolution from graphene to metals. We see, as we said before, 10 to 100 times higher signal resolution than metals. We're actually looking with metals and with graphene. So actually, this is a 60 contact interface made of graphene with bigger contacts and smaller contacts that you can almost not see. And what we did is compare it to an AD-Tech or a normal metal interface. What you see is that metals can see, for instance, in this case, beta bands, so frequency bands. We were talking before about the stopping band, between 10 and 30 hertz, and that's really common being observed by metals. But then when we look at graphene, the same dynamics, what we see is that there's a lot more activity in the brain. We see beta, of course, but we see gamma, even we see high frequency or single unit activity. And that's just by adding graphene or a new tool to understand what's going on in the brain. We have also seen that graphene has an unprecedented stimulation stability. As we said, we can inject 200 more charge injection, but at a very low impedance. Indeed, when we compare platinum, ignites, or graphene, and other polymers like PDOT, we saw that graphene has a similar stability over stimulation like platinum, but PDOT, for instance, was degrading and snapping in terms of impedance. And the last piece is that we've been able to actually read, of course, LFP, so the beta band, when it comes to a model of Parkinson's disease that we have now reproduced in rats, but also in other large animals. And what we were able to do is actually to look at this interspike interval distribution, which is mainly high frequency, and actually being able to dissociate a control from a Parkinsonian band, and by stimulating actually revert that band to a normal state, and therefore modulate that Parkinson or that abnormal tremor. So we are here with graphene to actually understand the brain better, understand the relationship between different circuits and different indications, and hopefully make a breakthrough when it comes to the progress of neurotechnology. And of course, we cannot do it alone. I mean, this is done with an incredible team, but also a very broad number of clinicians from all over the world. So you see here Canada, and you see here Berlin, Germany, but also California. And with the Nobel Prize of Graphene himself, that also joined our vision board. So I hope that in the future we could keep contributing to the breakthroughs in neurotechnology. Thank you.
Okay, thank you very much. We have time for one or two questions if you're both still available. Let me just say that I think I'd personally like to see interfacing graphene with even smaller structures like microtubules, which may actually be using phonons and electron excitons and phonons. So maybe we could talk about that offline. It'd be a slightly different application. Any questions? Dimitris.
Yeah, thank you for this very interesting talk. What are the high frequency oscillations like? How high can they be that you can decode? That's the first question. The other is, I mean, you're picking voltages, picking up voltages. So how is graphene? I guess it increases SNR somehow. But I'm saying compared to other materials you could be using, what is, let's say, the secret sauce there? Or I guess it's, again, higher SNR, some other properties. Maybe you can tell us a little bit more about that. Thanks.
So first of all, graphene is a very porous material. So just by this porosity, we have these kind of properties, the capacity to inject more charge, but also to have lower impedance. And to your point, when it's about high frequency, I mean, high frequency is always a debate. We have two kinds of graphene, graphene reduced oxide and CVD graphene. With CVD graphene and what we call transistors, we can read frequencies from minus 0.1 hertz to kilohertz. And with Ignite, we go from 500 hertz to kilohertz as well. So it depends in which model, and it's not always the case. I mean, we're learning every day, but yeah, we have seen it now in several species.
Thank you. Any other questions? I noticed in one of your papers, one of your colleagues' papers, you used graphene to measure up to maybe it was 100 hertz, but graphene can go much higher. And we've heard at this meeting evidence for detecting megahertz and gigahertz from the brain. Is that something you could look at with graphene?
we have not got there yet. But for sure, I mean, we see signals, we don't know what they mean yet. There's a lot that we still need to work on, and that's what we are doing. I mean, our objective is to get to first in human soon, and prove first that it's safe. I mean, that's always the question that everybody wants to clear. And we have progressed a lot there on the safety of the material, and of course the potential. And from the safety, we're very close, and I think we could say that graphene is safe from what we know today. And the potential is still to be fully explored. We are learning every day.
All right. Well, thank you both very much. That was great hearing from you both on this new frontier. And let's give them both a round of applause.
Thank you so much. Thank you, Zina.